Hi everyone, my name is Mireya Becerra Diaz. I am the local technical application scientist for BioLegend. First, I would like to thank the GRCF for organizing this symposium. In this seminar, I will talk about TotalSeq, a highly multiplex protein analysis in single-cell multiomics. But before starting, I want to let you know that BioLegend is constantly expanding the fluorophore catalog with new dyes such as Kiravia, Spark, and Fire dyes that will help you improve your flow cytometry analysis, either if you are working with conventional or spectral cytometry. Besides, we recently released updated new literature that you can easily download from our website. The outline for this talk is an introduction to single-cell RNA-seq, then integrating TotalSeq antibodies to single-cell sequencing, cell hashing, TotalSeq applications in CiteSeq, ending with showing you our portfolio expansion and other BioLegend tools that are important for enhancing your CiteSeq experiments. So let's start with the introduction. For those with no background in molecular biology, I want to give a little overview on what single-cell RNA-seq is. It is a technology that identifies single-cell RNA transcriptomics, allowing for a deep interrogation of cellular mRNA. With this technique, you can massively and in parallel measure thousands of cells, analyzing thousands of genes per each individual cell. It uses creative approaches, allowing for specific applications. You can have 3' or 5' directional sequencing, and you can also have BDJ sequencing for those that are interested in immunoprofiling. Let's talk about the workflow for single-cell RNA-seq. In single-cell RNA-seq, as the name indicates, what you want is to be able to analyze the transcriptome with a resolution of individual cells. For this, what you first need is to have a single-cell suspension. Then these single cells will be isolated in either individual droplets or microwells. This means that in the ideal scenario, you should have 1 cell per droplet or well. In this single cell suspension, you will also have 1 gel bead per droplet. Each cell gets a unique cell barcode, which is different in each bead. And each gel bead will also contain different capture sequences. Once in the droplet, the cells lyse and they release the RNA. Then this RNA interacts with capture sequences on the gel bead. And you can identify each RNA event because each capture oligo or sequence gets a unique molecular identifier or UMI. Then the next steps involve cDNA amplification, library construction, and sequencing. This way, we can map individual sequences to individual cells and have the expression of thousands of genes per cell, which is the cell transcriptome. So now let's talk about how to integrate protein analysis to single-cell RNA-seq. First, let's think about this question. Why could it be important to add protein analysis to single-cell RNA-seq? Analysis of cell surface molecules allows you to phenotype cells and identify different populations. Flow cytometry has been the gold standard technique in this regard. Although flow is great, when you have already identified the targets of your interest, it can run into caveats when you want to increase the number of proteins you want to analyze. Even the most capable cytometers won't allow you to measure more than 30 or 40 different proteins. And most importantly, with no or little simultaneous RNase data. But imagine the information you could obtain from your experiments if you could have both the protein and the transcriptome data. Let me tell you that having both protein and transcriptome data from the same individual cells is possible, and that it only requires staining the cells in similar fashion to flow cytometry before moving into sequencing. A recent publication out of the New York Genome Center used antibodies coupled with oligonucleotides in a method that they call cellular indexing of transcriptomes and epitopes by sequencing, or CytSeq. This method combines highly multiplex protein marker detection with transcriptome profiling for thousands of single cells. The method is compatible with two high-throughput platforms such as DropSeq and 10x Genomics. Originally, the antibodies were conjugated to streptavidin, and biotinylated oligonucleotides were used to link the antibody and the oligonucleotide together. But now BioLegend has a line of antibodies for CytSeq. These are our TotalSeq antibodies, which have the oligonucleotide already linked to the antibodies, allowing for an easy and fast one-step stain. As you can see in this t-SNE plot, having the single-cell transcriptome is helpful to identify different cell populations. But still, there are things that could be improved. If besides you add protein expression, the resolution of the differences between populations will increase, allowing to a more neat identification of your different cells and helping to identify, when this is the case, even subpopulations. I would like you to focus, for example, in the CD8 population here, that it's difficult to separate from the CD4. But when you have both RNA and protein information, this population is really nice result. Another case could be the CD16 monocyte population, difficult to separate from the CD14 monocytes. And here is really nice result and separate. So how is adding TotalSeq antibodies impacting your single cell protocol? As mentioned before, what you need to do is a cell stain in similar fashion to flow cytometry. Of course, the conditions are a little different, mainly in the washing steps. In CytSeq, the washers use a larger volume compared to flow. Also, viability is critical in CytSeq, but the incubation part, it's very similar. Once you make the stain and wash your cells, the cells can move into single cell droplet encapsulation and downstream sequencing steps. The thing to highlight here is that at the end, you will obtain two independent libraries, one coming from the mRNA and one from the antibody-derived tags or ADTs. So it was a lot of information. Let's make a summary and focus on the key takeaways. First, the TotalSeq antibody oligotags are captured similarly to mRNA. You will have an independent antibody-derived tag library. The ADTs and the cDNA library will share the same cell barcode. And each oligo will have a unique molecular identifier or UMI. These characteristics will allow you to map individual sequences to individual cells. Now let's move into some technical validations. In this slide, what I want to show you is the analysis of surface proteins in human cells when using CytSeq and in comparison to flow and CyTOF, two gold standards for surface protein analysis. In this first comparison, you can appreciate the expression of CD19 and CD3 when using flow and CytSeq antibodies. In this second example, you can see the expression of CD4 and CD3 comparing CyTOF and CytSeq. As you can see in both comparisons, CytSeq antibodies provide a similar or even improved resolution of the cell populations. This demonstrates that CytSeq antibodies are as efficient as those used in the gold standards. Moreover, having the protein expression besides the transcriptome would help decreasing challenges such as RNA dropout and the divergence between RNA and protein expression. It is known that mRNA levels do not always correlate with protein expression. You can find these nicely visualized in these dot plots, showing CD14 and CD8 protein and gene expression. In this plot, CD8 protein is nicely expressed, but not the CD8 gene. The other way around in this other plot, the mRNA for CD14 is nicely expressed, but the protein signal is not that strong. And in this graph, that shows ratios of cells positive for protein and negative for gene expression in the gray part of the bars, or positive for both in the purple part. You can see that gene and protein expression are not always 100% parallel. So having both transcriptome and surface protein expression profile will provide a much more accurate understanding on the cell activation and function. Now let's give a look at the different TotalSeq antibody formats and their compatibilities. We offer TotalSeq antibodies in 4 different formats. And the differences between TotalSeq A, B, C, and D are due to different oligonucleotides. Each distinct oligonucleotide provides compatibility with single cell partitioning platforms. So the first one is TotalSeq A antibodies. TotalSeq A antibody oligos contain a 3' poly-A tail that can be captured by platforms utilizing poly-DT capture sequences. Then TotalSeq A antibodies are compatible with any droplet-based single-cell platform. Then we have TotalSeq B antibodies. These antibodies are compatible with 10x genomics single-cell 3' kits. The capture of oligos in this case does not depend on the poly-DT sequence, but instead it relies on a specific capture sequence present in the genomic, in the 10x genomics gel bead. Then we have TotalSeq C antibodies that are formulated to work specifically with 10x single cell 5' immune profiling kits. Capture of TotalSeq C oligos is dependent on the template switch oligo or TSO found in the 10x immune profiling beads. The TSO adds a common 5' sequence to full-length cDNA that is used for downstream cDNA amplification. And last, I want to introduce TotalSeq D. These antibodies work in a completely different platform that does not measure any RNA at all, and that specifically measures DNA. TotalSeq D antibodies are formatted to work specifically with a Mission Bio tapestry system. Capture of the TotalSeq D oligo is achieved via biotin streptavidin dependent on a common sequence that targets single-cell DNA and protein profiling. So let's summarize this part. TotalSeq antibodies use a similar staining protocol when compared to staining cells for flow cytometry. The antibody-derived tags are specific to antibody targets. CytSeq shows a similar or superior performance to flow and CytELF. CytSeq can reveal different patterns between RNA and protein. Antibody tags capture method is similar to that of mRNA, and an independent antibody-derived tag library is constructed. All this makes TotalSeq a seamless and integrative solution into existing single-cell protocols for simultaneous characterization of protein and RNA or DNA. Now let's talk about something that I am sure you will like. These are our hashtags antibodies. So what is cell hashing? It is a way to barcode different samples in order to be able to combine them into a single lane, and then the multiplex during the analysis. For cell hashing, we have our hashtag antibodies. These antibodies label cells by targeting ubiquitous proteins. Hashing provides some important advantages, as you can fit multiple samples coming from different experimental groups into the same run. You can, using the hashtags, decrease the variability among experimental conditions and also decrease batch effects. So what are the characteristics of these hashing antibodies? Well, the antibodies target ubiquitous proteins. For the human version, they will target CD298 and beta-2 microglobulin. And the mouse hashtags will target CD45 and H2MHC class 1. As mentioned previously, cell hashing allows you to combine samples within one single cell RNA-seq run. This is called sample multiplexing. A very convenient thing is that our conjugates are premixed and ready to use. Hashing your samples will help with data cleansing, and I will show you how in the next slide. Moreover, hashing your cells will allow you to superload the 10x lane, loading up to 20,000 cells per well. Let's look at the hashtags in a more schematic way. In this example, you can see 3 different samples from 3 different donor source conditions that are stained, each one with a different hashtag antibody. After washing the samples, they were pulled together on a single cell 10x 3' lane. The samples were processed, the libraries prepared and sequenced. Then you can demultiplex your next generation sequencing data to identify each different sample based on the different hashtags. And here is where I want to show you how hashtags can help you identifying droplets containing more than one cell, and then these reads can be excluded of your analysis, helping cleaning your data. So now let's look at an example. In this report from John Ashton, they worked with 2 acute myeloid leukemia patients. Bone marrow samples were collected at the moment of diagnosis and relapse. Once all samples were collected, the cells were hashed and sorted for CD34. Then the cells were pooled and 10,000 cells were loaded per well and sequencing was performed. In this Disney plot, you can see the 4 different samples together. The question you may have at this moment, after I mentioned that CD34 cells were sorted, is if sorting may affect the hashtag signal. Well, here you can see how cells look if you stain them prior or post-sorting. Nice thing to highlight is that the hashtag antibodies are compatible with sorting with no or minimal loss of antibody occurring during cell sorting, as you can observe in here. Now let's see how samples can be resolved after hashing and pooling your cells. As you can see, patient 1 and 2 have different phenotypes in certain leukemia proliferation assays. The important thing to highlight is that you can demultiplex in such an effective manner that you can identify these differences between samples then run in the same lane. And this group is now currently working on expanding the data with some refractory and remission samples using the hashtags. I think it's time for another summary. First, hashtags can be used to identify sample origin. They will allow you to multiplex separate samples in the same 10x lane and to superload the 10x lane with up to 20,000 cells. This will help to decrease batch effects and variability among experiments. And of course, it will also help to optimize your budget. The conjugates are already premixed and ready to use and are available in all three formats, TotalSeq A, B, and C. And to make this more graphic, here in this business, you can see how 8 different samples were pulled and multiplexed, and how nicely these 8 populations are resolved, and how droplets, the little dots here, containing more than one cell, can be identified and excluded from your analysis. So how has been the evolution of TotalSeq antibodies and the multiplexing capacity? As I mentioned before, this story started just a few years ago in 2017, when a cocktail of 14 antibodies was used. Later in the same year, up to 82 different antibodies were used in combination. And at this moment, BioLegend has used up to 220 different antibodies in one single stain. As part of this project of the 220 antibodies, some PBMCs were stained with all these antibodies in the TotalSeq A format. After sequencing, cells were clustered based on either 10 of these antibodies, 9 of those correspond to our TBNK cell panel, or with a whole 220 antibodies. And in the next slides, I want to show you how cell populations look when you use 10 or 220 different antibody to identify the cell populations. But first, I want to introduce you to the TBNK cell panel. This cocktail contains these 9 antibodies. It comes as a single reaction vial and has been titrated on several sample times for its optimal staining. These antibodies are lyophilized for increasing the stability, and having these antibodies as a cocktail also helps to reduce cost and make the cocktail very easy to use. Based on these 9 antibodies, you can identify different populations, and I want you to focus in the CD8 positive population here in this red circle. But what happens when, instead of analyzing your cells based on 9 or different antibodies, you make the analysis based on the whole 220 antibody cocktail? Well, what was first looking as a solid single population resulted to contain different subpopulations based on the differential expression of these antibodies. Don't worry, I won't describe the differential expression, the differential protein expression of each different subpopulation. I just want to show you that they were able to identify 6 different subpopulations contained in what was just a solid CD8 population. Just to mention, the different subpopulations were naive T cells, T central memory cells, effector memory T cells, effector memory positive for RA, gamma delta T cells, and mucosal associated invariant T cells. And the message I want to share here is that the ability of analyzing more markers helped to uncover different subpopulations contained in what it looked as a uniform single population. Now it's time to look at our portfolio and the different panels that BioLegend offers. You may remember that TotalSeq antibodies come in 4 different formats: TotalSeq A, B, C, and D, where TotalSeq A, B, and C can be paired with single-cell RNA-seq, and TotalSeq D can be paired with single-cell DNA-seq profiling. In our catalog, we have anti-mouse and anti-human antibodies, individual antibodies in all different formats. We also have predefined cocktails. We have the human cocktails in all formats, A, B, C, and D, and yeah, A, B, C, and D. And recently, BioLegend launched the first anti-mouse cocktail in a TotalSeq A format. And new anti-mouse cocktails for TotalSeq C and B are expected to be released during this 2022. So now let's look at the different antibodies. This slide may be a little bit overwhelming, but what I want you to have is an overview of all the different options that BioLegend has for TotalSeq. Here it's the portfolio for TotalSeq A. We have individual anti-mouse antibodies, anti-mouse hashtags, anti-human antibodies, anti-human hashtags. The cocktails, the universal cocktails in human, and the TBNK, the ones you saw recently, and the new TotalSeq A mouse universal cocktail. What I want to highlight is, or I want you to focus in the number of antibodies contained per cocktail. In this case, the human universal cocktail contains 154 different antibodies and 119 for the mouse version. Similarly, we have a variety of antibodies for TotalSeq B, very similar to TotalSeq A, just missing the mouse cocktail, and TotalSeq C. Again, just to highlight the number of antibodies contained in the different cocktails, mostly in the universal cocktails. We also have TotalSeq D. In this case, we only have antibodies against human. We have individual antibodies and the human heme oncology cocktail containing 42 different antibodies. And also for those interested in profiling gene expression in cells that are difficult to isolate and use single nuclei RNA sequencing, BioLegend offers a line of TotalSeq nuclear hashtags in TotalSeq A and B formats. In both cases, this hashtag target the nuclear pore complex proteins. So as you have seen, our panels, particularly the universal cocktails, contain a large number of antibodies. However, it may happen that you would like to have, besides the targets contained in the cocktail, another antibody that is not present in the cocktail. These antibodies could be from our catalog or a particular antibody that your lab is interested. And let me make a parenthesis here. At BioLegend, we offer custom conjugations for TotalSeq antibodies for all the different formats. These conjugations could be done on purified antibodies from our catalog or from a different source. You can provide the antibodies. And in this second case, we just need to check purity and some other characteristics as the buffers they are contained in. But custom conjugations can be done. So yes, new antibodies can be added to a predefined panel. And this is what we call spiking. And you can find the antibody cocktail spiking guidance in our website. So if you are interested in adding a specific peculiar antibody to your panel, this can be done. As mentioned before, our cocktails are pre-titrated using optimal concentrations for staining. However, when working with individual antibodies or spiking in new antibodies to a cocktail, you need to determine how much antibody you have to add to your samples. Same case when you are using the hashtags. Of course, there are recommended ranges, but the ideal is that you titrate your antibodies in your particular samples and experimental conditions. For this, we recommend performing titration via flow cytometry. This means that you can use PE or APC conjugates of the same antibody clones that you are interested in titrate and perform some titration curves. We recommend using 5 to 8 different points of titration series for each antibody, as shows in these histograms. And you can find a blog talking about tips and tricks for titrating TotalSeq antibodies and more resources in our website. Now it's time for another summary. So let's see the panel key takeaways. The panels provide convenience with single reaction vials. These vials are pre-titrated using optimal concentrations for staining. Something you will like is that using a cocktail will reduce cost compared to buying individual antibodies. Panels come lyophilized for stability. Also, using the panels will minimize variability between experiments, between different labs, and during longitudinal studies. And the panels can be combined with BioLegend hashtags for sample multiplexing. Another thing I want to talk about is the MojoSort dead cell removal kit. So running dead cells during site-seq essentially leads to wasting single-cell runs on dead cells, which may result in suboptimal data or not processing enough cells to pick up the positive events, especially if the frequency of your target cells is very low. Also, that or damaged cells can incorporate antibodies in a non-specific manner, resulting in false positive signals or very bad background. Thus, we recommend to use your samples with a viability above 95% before beginning your site-seq experiments. But what to do if your sample has less than 95% viability? Well, there are some commercial kits that can be used to remove dead cells. At BioLegend, we offer our MojoSort dead cell removal kit. As all our MojoSort kits, it is a magnet bead-based assay. In general, MojoSort uses magnetic beads for cell enrichment, which could be either positive or negative selection. In this case, it's going to be a negative selection. MojoSort dead cell removal kit utilizes a complex formed between apomonomer and streptavidin nanobase. This complex will bind to phosphatidylserine residues exposed on the surface of apoptotic and dead cells with high affinity. You incubate your cells with this mix, and after a quick wash, you proceed to pull out the dead cells using the MojoSort magnet. And then the untouched live cells can be collected by decanting the liquid into a fresh tube. Something important to mention is that this kit does not need any specialized buffer containing high calcium concentrations. So, what's the performance of this kit? Here we have representative data from both mouse and human dead cell removal kits. We use Helix Blue and ApoTracker Green to show the dead cells in both mouse and human samples. For mouse, we use heat-killed splenocytes, and for testing the human kit, we use heat-killed PBMCs. You can see that viability started in both cases around 65%. And after pulling out dead cells using the MojoSort dead cell removal kit, live cells were enriched and viability was increased to around 95%. And all this was done in standard cell separation buffer. Another good thing about this kit is that not only enriches live cells, but it has a superior live cell recovery when compared to other similar kits that require high calcium concentrations. In these graphs, we can appreciate how using BioLegend's MojoSort dead cell removal kit resulted in a greater percentage of live cell yield for both cell types, human or mouse. Again, with the advantage of no no need for a high concentration of calcium in the buffer. So how does removing dead cells would actually impact your sample quality and data? Let's look at this example using human PBMCs. Here we have a site-seq experiment containing fresh, 60% live, where we added back some heat-killed PBMCs, and same 60% live sample where dead cells were removed using our kit prior to experiment. Site-seq was performed on all samples, and the mitochondrial reads are shown here as a correlation with dead cell. Sorry, with cell dead. As you can see, there is a higher percentage of mitochondrial reads in the 60% live group, while the module dead removed group looks very similar to the fresh cell group. Also, noting this UMAP plot, we can see that first, common PBMC population can be detected in all samples. But in the dead cell population, we mostly find dead cells from the 60% live group. As you can see, it's all blue. While there is an absence of dead population in the fresh and in the dead removed samples. So there is almost no green or orange in this dead cell population. Here is just another way to visualize that the dead cell population has a greater percentage of mitochondrial reads. And the last thing I want to talk about is BenSeq. BenSeq, this acronym comes from bulk epitope and nucleic acid sequencing. And the main difference with single cell seq is that in BenSeq, the RNA analysis is done in bulk, or in other words, on the whole sample and not on individual cells. BenSeq is actually a predecessor of single cell RNA-seq. It provides with an average of transcriptome for a sample. This is a great approach when you are looking for a differential gene expression, usually comparing experimental groups that are clearly different, such as control and treated, or control and disease. And most important, BioLegend has validated TotalSeq A antibodies to be compatible with BenSeq. Similarly to what happens in site-seq, in BenSeq, you will also have at the end of your sequencing two independent libraries, one coming from the ADTs and one from the mRNA. As you can see, BenSeq provides a cost-effective way of adding proteomics data to RNA-seq experiments, as it is relatively inexpensive compared to single-cell RNA-seq, because it does not require any single-cell capture instrumentation or reagent cost involved. If you are interested in learning more about TotalSeq or site-seq in general, I would like to invite you to visit these websites or give a look at these publications. Just let me know, and I'm happy to share this information with you. Also, I want to acknowledge our collaborators, external and also to the internal proteogenomics team. And last, I want to thank you all for attending. Also, please come visit the BioLegend virtual booth. And now I am happy to take any questions or comments.